Okay, so I am going to be going over on this video topic 20, which in your book is page 35. Let me just double check that. Yeah, okay. And this is looking at structures of the eukaryotic cell. I'm not sure why. I'll have to go back to prokaryotic versus eukaryotic. We'll do that in person. So this should all actually be review material. And it's going to start with the plant cell, and then it's going to go on to the animal cell. So let me get this presenting. So this looks similar to the picture that's on topic 20, page 35 is the page. And we'll tackle the plant cell parts first. And so you'll just kind of take notes with me here. The first structure that they have labeled structure 1 is the chloroplast. And what we need to know about the chloroplast is that it has two membranes. And in a plant cell, yes, there's going to be many of these per plant cell. They're going to be green in color because they contain the pigment chlorophyll. So that's what makes them green. And you should realize that them being green means that they are going to be absorbing every other color of light and reflecting green. So that's going to be important when we talk about photosynthesis. There's two membranes. So there's an outer membrane and inner membranes. The inner membranes, we need to know that these are called thylakoids. I apologize, it's a little noisy in the hallway. And these are the sites of light gathering. So you want to think of these kind of like solar panels. They're going to gather light energy. So those are called the thylakoid membranes. They're actually usually going to also have like little, I'm doing this in green, but that's not that easy to see that, connections between them. So they look more like this. Actually, there's connections between the different stacks. So these are called thylakoids. And I don't have to write it here. You could see it right here, but just to make sure you know that, they're thylakoid. The thylakoid membranes, those are going to be the sites of light gathering for plant cells in the chloroplast. And then this fluid-filled portion here, this is called stroma. And stroma is the fluid. This is going to be the site of the Calvin cycle, which we're going to learn all the details of. But if you can remember right now, Calvin cycle takes CO2 and converts it into carbohydrates. Those four C's. Calvin cycle, CO2 converts it into carbohydrates. And so overall, obviously, the function of the chloroplast is photosynthesis. Sorry. Okay. And then the next structure is on this page 35, topic 20, is labeled structure 2. This is the nucleus. And what you should realize about the nucleus is it has a double membrane, and in that double membrane, there are pores. I don't think we can really appreciate the pores on this particular nuclear cartoon, but there's pores that things can go in and out of the nucleus. So things like mRNA can come out of the nucleus. Other things can come into the nucleus for signaling. The nucleus is going to contain the DNA and the RNA. And actually, also, there's a structure that also stains usually that's inside of the. Oh, is this the, yeah, this little purple part here that would be inside of the nucleus that is actually what, when ribosomes are forming, that's like what the nucleolus actually is the ribosome parts that just are a little bit more dense inside the nucleus. And so the reason I have a question mark here about the nuclear, I don't know if I can move this. That flips it. That's full screen. We don't want that. I can close it. So you don't need to look at me anyways. You just need to listen to me. All right. So frequently we say that the nucleus is the control center of the cell because that's where all the blueprints, the instructions are. We get those instructions out of the nucleus and we protect the nucleus in there. But there's also a lot of other parts of the cell that also signal. So it doesn't mean that everything that happens in the cell has to go through the nucleus, but a lot of things do. All right. So I think that's enough for the nucleus for now. And the structure that's labeled 3 on your diagram looks like it comes right off the nucleus. And so we don't really see that in my little cartoons here, but that is and you do actually see it nicely up on this one and the one in your book. But that is the rough endoplasmic reticulum. Endoplasmic means inside the cytoplasm, and reticulum means little network. So structurally, the rough endoplasmic reticulum, this is the rough one. You can see that it's studded with these ribosomes, and it's like a passageway or tunnels. So they're all connected with each other, and they are studded with ribosomes. So their function is, the ribosome's function is protein synthesis. But when the protein synthesis happens right into the tubes that are going to take the protein where it needs to go, then that's the job of the endoplasmic reticulum. So it's going to transport protein around the cell for the rough endoplasmic reticulum. And I've seen different estimates, anywhere from 20 to 70% of certain cells. If the cell has to make a lot of protein, like for example in muscle cells, then it's going to have more rough endoplasmic reticulum to be able to transport that around the cell. Okay. So that's the rough endoplasmic reticulum. Next is the smooth. And so this is referring to this picture is showing a smooth endoplasmic reticulum. I think that the smooth endoplasmic reticulum, if I had to pick an organelle, I would probably pick the smooth organelle, a smooth endoplasmic reticulum to be my favorite organelle. It's the membranous tunnels of the smooth endoplasmic reticulum are more tubular, so you can appreciate that. The rough ER is more flattened, and then the smooth ER is more tubular. The smooth ER functions to detoxify things. It also functions for lipid synthesis and calcium storage. So if we think about this, and I've told this story before, if you've had me in biology, I know I say that a lot. And if somebody needs to detoxify a lot, let's say that they are on medications that are processed through detoxification by the liver, for example, um, or in the case of alcoholics, that the liver is going to detoxify a lot, they're going to need more smooth ER. And the one of the ways to get at that is to convert some of your rough ER to smooth ER. And the problem with that is, while you get more detoxification, that's what tolerance is. That builds up tolerance. So more and more of the substance are needed to have the same effect on the body. And if you have more smooth ER, yes, you get more detoxification, but look at the other functions of the smooth ER. It's also involved in synthesizing lipids, so you can end up with a fattier liver. So if you replace liver tissue with fat, that is a bad thing. And same thing with calcium. So calcium, if you have too much calcium, just like it would be like it hardens our bones, it would harden the cells, it would harden the liver cells. So that's called cirrhosis of the liver. And yeah, so that's a little bit about the smooth ER. All right, next structure on your page is structure 5, which is a ribosome. And the ribosomes are made of protein themselves, and they have two subunits. So, like, there's a larger subunit and a smaller subunit. And they also have ribosomal RNA, and that also helps with their structure. And they can either be freely floating around in the cytoplasm if the material doesn't have to be transported, if it's a cytoplasmic protein, or if it needs to be transported somewhere else in the cell, then the ribosomes that are studded on the rough endoplasmic reticulum take care of that. I don't know how they differentiate that, but and so just remember that we go from DNA to RNA to protein. So what we see in this picture, it's a still image, but this long strand thing here is going to be the mRNA. It's going to read the ribosome. It's like a protein making factory. It's going to read that, bring in the correct amino acid, and link it up with this growing polypeptide chain in the case of, and we see tRNAs in the background here as well. And so actually on this tRNA, you can see it nicely. That red part is the amino acid that one is going to be bringing in, I believe. And so this is a really long amino acid chain or protein. And that, of course, that process is called translation. Again, this should all be review. All right, next structure 6 on your diagram is the Golgi apparatus. And the Golgi apparatus, there's a few per cell and present in all eukaryotic cells. They look like you took like a stack of pancakes and you pinched them down the middle. And so they have a little bit of wider openings on the ends. And Golgi complex trafficking signaling is very complex, still a lot of questions. But in general, the Golgi apparatus is going to add sugar tags to process different material like proteins and lipids and give them their destination inside and outside the cell. So the analogy of Golgi being the post office of the cell is actually a very good one. All right. Now, I think that's all the structures that are on page 35. And then if we flip the page over, we see kind of like a classic animal cell diagram, which I thought I had something for this, but I'm still getting used to Canva. And so when the structure number 7, I didn't number these, 7, 8, 9, I think. So centrioles are present in just in animal cells. So plant cells do not have distinct centrioles, although they do still have to have microtubules to be able to pull apart their chromosomes during cell division. That's what the microtubules do. I mean, that's what the centrioles do. But so plant cells, they just have, it's not as organized, but they call that like a MTOC or microtubule organizational center. So it's just not as distinct of a structure. And it looks like they took like, it would probably be pretty decently easy to make a model of a centriole because you have these two structures that are at right angles. I try to orient these as best I could to that orientation. And they look like you have like 3 straws that are kind of glued together. And then there's going to be 9 of those triplets. And so those triplets, what I'm talking about are microtubules. And this is what brings this is what pulls apart the chromosomes during cell division. Again, only in animal cells. Something similar in plant cells, but it's not as distinct and organized. All right, I want to kind of clear up a little bit of misconception here. Hopefully, you don't still have this misconception, but the mitochondria are present in all eukaryotic cells, not all cells, but all eukaryotic cells, plants and animals, and fungi and protists. So I should have added those to that as well. But a lot of times people think, well, plants have chloroplasts for energy and animals have mitochondria for energy, but plants also have mitochondria that they use to break down the stored sugar that their cells made by photosynthesis. So they are, if you look on the previous page, you do see mitochondria. They just reserve the labeling of them on the animal cells, which I don't think helps to clear up that misconception, but it is what it is. So all eukaryotic cells have mitochondria. And we're going to study the mitochondria in more detail. I probably want to do that in person with you, but if we can get started with it, all the more better. So the mitochondria consists of an outer membrane, so that's labeled there, or the outer membrane is here. It has an inner membrane, and that inner membrane, this is going to be very important. We're going to keep coming back to this and talking about it over and over and over again. Those folds are called cristae, and inside of the inner mitochondrial membrane, and I'm not going to draw all these, but it is completely studded with enzymes that help synthesize ATP, as well as these big protein complexes that transport electrons in the mitochondria to eventually produce ATP energy. So, yes, it's the powerhouse of the cell, but please take it, we're AP biology, so we need to step that explanation up a little bit. So, powerhouse of the cell means to make lots of ATP using oxygen as a final electron acceptor in something called an electron transport chain. And that's a lot right now, but we're going to keep coming back to it. We're going to do a whole chapter on energy and photosynthesis and cellular respiration. All right, so I think that's good for the mitochondria. And then the lysosome is, I say, a vesicular structure. What I mean by that is it's circular, and it's so vesicle means a circular structure, and in lysosomes contain a lot of digestive enzymes, different types of digestive enzymes. And one example that I like to talk about is genetic disorder called Tay-Sachs disease, which is a recessive genetic disorder. And I want you to watch that video that I have posted on Google Classroom under this assignment as well, called One Wrong Letter. It's about Tay-Sachs, and then also answer questions 10 through 12. And have a good weekend. And I will see you on Monday.